Hi and Assalamualaikum everyone. My name is Nur Afika binti Arjunaidi, and today I will present to you about the novel drug development for type 2 diabetes. Okay, so this is the flow that I will follow and explain each of them during this presentation. Firstly, I would like to briefly explain what type 2 diabetes is. So, type 2 diabetes is also known as non-independent diabetes mellitus or NIDDM. So, this disease usually occurs in elderly group of people, especially those who are overweight and have an unhealthy lifestyle. And people with type 2 diabetes develop resistance to the secreted insulin. And type 2 diabetes is the predominant form of diabetes and contributes to 90% of people with diabetes around the world. And there are a range of drugs like sulfonylurea, biguanides, glinides, and glitazone that are used for the treatment of type 2 diabetes, but all of them have an unintended effects like hypoglycemia and obesity. So as the chief scientist in Prime Pharma, I would like to develop a drug with less side effects. One of the most important steps in developing a new drug is target identification and validation. So a target is a broad term which can be applied to a range of biological entities, which may include, for example, proteins, genes, and RNA. So a good target needs to be efficacious, safe, meet clinical and commercial needs, and above all, it must be druggable. So, the target that has been chosen is the peroxisome proliferator activated receptor gamma or PPAR gamma. So, PPAR gamma is a nuclear receptor which is highly expressed in the fatty tissues and in skeletal muscle cell. The PPAR gamma agonist will act by facilitates the insulin sensitivity to enable maximum glucose uptake in adipose tissue and also maintain the body homeostasis. Okay, next, once the target has been identified, if then it needs to be fully prosecuted or validated. So, validation technique range from in vitro tools through the use of whole animal models to modulation of a desired target in disease patients. And recently, chemical genomics, which is a systemic application of two molecules to target identification and validation, has emerged. So, chemical genomics is the study of genomic responses to chemical compounds. And the purpose of chemical genomics is to provide a chemical tools against every protein encoded by the genome. So, target validation can be done through chemical genomics. The next phase is the discovery of hit and lead molecules. The strategy that I plan to obtain the lead molecule is through CADD or computer-aided drug designs. So once a promising drug target has been identified, researchers will aim to identify molecules that can interact with the target to produce desired biological effects. So firstly, we have to identify the hit molecule. Hit molecule is a compound that have the desired activity in a screening assays, and it can be done through high throughput screening or HTS. And it is important to develop pharmacologically relevant screening assays for hit discovery and for the subsequent hit to lead selection process. And then, after several hit compounds have been identified using high throughput screening or HTS, lead compounds need to be identified. So, lead compound or lead molecule are chosen from the collection of hit molecules which have the most desired activity through several screening methods. And it can be done through computer-aided drug design. Okay, so what is CADD? CADD or computer-aided drug design is a method that involves the use of a high-performance computing to find the most possible drug candidate. Under CADD, we have two types. which are structure-based drug design and ligand-based drug designs. So as we already know the structure of the PPAR gamma receptor, we choose the structure-based drug designs. So structure-based drug design relies on the knowledge or idea of the 3D structure of the target, which can be either obtained through NMR spectroscopy or X-ray crystallography. So, using this structure, the candidate drugs that are predicted to bind with high affinity can be determined and observed through molecular docking and scoring. And moreover, there are some advantages of CADD over other methods, which include lower cost, and it reduces the amount of chemical weights used, and helps to improve to understand the drug receptor interactions. Okay, next I will explain about molecular docking. Molecular docking is a method which involves the prediction of the preferred orientation of one molecule to a second, especially during binding process, in order to form a stable compound. And as we can see in the figure here, the ligand and the target have bind together and form receptor ligand binding models, and are expected to have high affinity. So the affinity can be measured by using scoring. So scoring method is used to measure the binding affinity between the two molecules. Okay, next, after molecular docking, we have to do molecular dynamics. Molecular dynamics is a computer simulation method that is used to study the physical movement of atoms and molecules. So the atoms and molecules are permitted to interact for a fixed period of times, and will give a view of the dynamic evolution of the system. This is important in order to observe truly on how the binding occur. So before we proceed with clinical trials, we have to do lead optimization, and this method will be done after we have find the right lead molecule. So lead optimization. The goal of lead optimization is to maintain the favorable properties of the lead compounds and improve the imperfection or disabilities of the lead compound properties. So the properties include ADME, absorption, distribution, metabolism and excretion, toxicity, ionization, and also water solubility. And the improvement can be made by making a small modification to the lead compound through structure activity relationship or SAR. Okay, so after we have done with lead optimization, we can proceed with preclinical trials and also clinical trials. Okay, so these are all my references that I referred to do this drug development, and that's all from me. Thank you.